Hi all, how are you doing today? So I guess this is the last meeting of our section. So welcome all. It's my pleasure to introduce Yves Smet. So Yves has received his PhD at the University of Leeds, and then he moved to his very first postdoc at Tübingen, as far as I remember, right? Germany, in the Max Planck Institute of Developmental Biology. And then he moved for his very second postdoc at Ghent University, maybe as a preparation for holding group leader position at the VIB, PSB. So Yves, many thanks for coming here for our section, and I guess the stage is yours, man.
Okay, good. Thanks for the invitation. And I'm kind of hopeful that I can whiz you through how we are trying to unravel temperature-mediated signaling in plants using phosphoproteomics and trying to build signaling networks. So, I mean, it's probably unnecessary to put this into context because everybody is very much aware that temperature is rising over the past years, past decades. And of course, this rise in temperature, global temperature, has an impact on our crop yield. And this is an example from wheat, where you basically see that per degree increase above the optimal temperature, there is about a 6-degree decrease in overall yield. So we really must urgently try to understand how plants deal with this and what the underlying molecular mechanisms are that are associated with temperature signaling and temperature responses. So we have been looking at this in a number of ways in plants, both in the model plant Arabidopsis, where there are a number of typical responses that are associated with these kind of mild increases in temperatures. It's kind of a warm temperature. It's not an extreme heat stress that is ultimately leading to lethality, but we're really looking at this kind of minor increases of about 5 to 6 degrees that have an impact on the plant with, for example, the lengthening of the hypocotyl, the lengthening also of the petioles. And this is furthermore associated with the rising of the leaf, so kind of a changing in leaf angle from this kind of more flat position to a more upward position, which can also be seen here in these soil-grown seedlings. In addition to that, plants at elevated temperature also try to cool down because this change in architecture is a cooling response, so it kind of allows an improved airflow through the shoots. But in addition to this, an additional cooling mechanism is, of course, stomatal opening. So you can see that at elevated temperatures, more stomata will open, and this kind of will also lead to an additional cooling of the leaves. So we've been trying to explore this not only in model plant Arabidopsis, but also in a number of crops, such as wheat. And you can see here that this kind of lengthening of the organs along this temperature gradient is also nicely visible with these kind of wheat leaves. So if you have a more or less optimal temperature around 19 to 24 degrees, if you increase that gradually, you see that organs will become longer. If you further increase the temperature, there is again a shortening, a kind of a reduction in growth. If you go even beyond that, this will lead to yellowing of the seedlings and ultimately lethality. And of course, also in monocots such as wheat, stomatal opening is triggered at elevated temperature. So we're trying to tackle this from a kind of early stress perception, early signaling kind of point of view, where we impose a temperature stress, and then we're really trying to capture what is happening very much in the beginning following this stress signal. And we are zooming in especially on signaling events associated with protein phosphorylation, as this is a reversible post-translational modification on a limited number of amino acids mediated by kinases and phosphatases. And by exploring this, we are really trying to find what is happening on the molecular level and also what is happening there very early on. And when we embarked on this project now a kind of number of years ago, very little was known about post-translational modifications, especially phosphorylation, in the context of these growth responses, which are collectively also referred to as thermomorphogenesis. So there were a number of elements that had been shown, where of course there are a number of key components, key temperature sensors, such as, for example, Phi-B, that play an important role, that impact on a very essential transcription factor, PIF4. These factors are also, in addition to their role in temperature, involved in light signaling. And in addition to that, there are a number of kinases, phosphatases, that were shown to play a role in the context of brasinosteroid signaling that really impact on these responses as well. And then recently, and this is what I will talk about in my presentation, we were able to add this module here that controls early signaling responses associated with these changes. So one of the ways we address this, and this is one of the goals in my group, is that we are trying to look for conserved signaling components. So we kind of try to identify factors that play a role not only in Arabidopsis or not only in wheat, but that are conserved across multiple plant species. So coming back to this temperature gradient, where I showed this different growth profile, we also grew plants at different temperatures for 60 minutes. We kind of exposed them for 60 minutes to different temperatures, and then we captured the phosphoproteome. So we basically use the standard workflow where we extract proteins, enrich for phosphorylated peptides, and then subject these to LC-MS/MS to then really see what is differentially regulated at these different temperatures. And there we observed that indeed you get distinct groupings in this UMAP plot, which is kind of similar to a PCA plot, kind of trying to reduce the multidimensional complexity into 2 dimensions associated with these different temperatures. And there is also a group of peptides that doesn't seem to really obviously respond. So if we then look a little bit closer at our data and try to group these different temperatures together, we kind of observe 3 dominant responses. On the one hand, the 14-19 degree response, and this is also reflected here in this correlation map, the response to 24 and 29 degrees, and then 34 degrees. So we have basically across the gradient 3 distinct molecular signatures that are associated with temperature. So we then zoomed in into these data, we kind of looked at all our identified phosphosites. We focused on those which have a localization probability above 0.75. We kind of filtered for a limited number of missing values. And then we identified about 226 highly significantly temperature regulated phosphosites, which we then explored in a little bit more detail. So one of the things that we did, because we had this temperature gradient, and we considered this gradient similar as a time series. So basically, every temperature in this case is a time point, so to say. And we use this gradient information to build signaling networks. And this kind of, and we used a tool that we developed together with the lab of Ross Sassani, called NetForce, where we are basically able to pinpoint different kinases associated that peak at different temperatures, and then we can link these or predict what their putative substrates would be based on their phosphorylation profiles across this gradient. And this allowed us to pinpoint a number of key kinases. And just this one is maybe something to remember, because I'll come back to that in a minute, TOI4 and TOI5. That seem to peak in this case at the slightly lower temperatures. But there are a number of other ones that peak at these intermediate and higher temperatures as well. So when we then looked a little bit closer at our data, we have these kind of obvious 2 clusters of up and down regulated phosphopeptides, which we then tried to further subcluster because we were interested in the fact, as I've kind of shown you earlier, you see along this gradient that there was this kind of maximum growth at some point, which is then going down again. We were interested if phosphorylation profiles would also follow that kind of bell shape along the gradient. And indeed, if we look at a number of clusters, we do see that along the gradient there are phosphopeptides that really follow this bell shape, either in a positive or in a kind of more opposite way. And these are, of course, some of the interesting candidates that could be associated with this kind of threshold response, this kind of tipping response. And at the moment, we are looking into this in a little bit more detail. The second thing that we were interested in is, can we use this temperature-regulated phosphor proteome to really identify specific variety-dependent molecular responses? So if you would take wheat varieties that have a known response to temperature, this either being sensitive or tolerant, can we also reveal this by looking at our phosphoproteome? And we exposed these 4 varieties. So our original data was done on cadenza, and then we added these 3 varieties with a distinct response, and we see that indeed they have a response to low and high temperature. And these 3 varieties tend to group a little bit more together than this cadenza variety. But then we use this information to really see if the responses would be similar or if there would be opposite responses in phosphorylation. So if we look at the tolerant versus the sensitive varieties, basically what you see plotted here is if there is an upregulation, it's indicated in red. If there is a downregulation in blue, and if there is no change, it's basically in gray. And each line represents a phosphosite. So basically we see here that these upregulated ones in the sensitive variety are to a large extent not regulated in the tolerant variety. And we see something similar with downregulated ones, which are then, for example, in this variety, not downregulated, but stay at the same level. And this kind of allowed us to really try and identify responses, molecular responses that are associated with either sensitivity or tolerance. And this could then be used potentially in breeding. And our preferred molecular markers in this case are those that in the sensitive variety are nicely upregulated, that do not change in the tolerant variety, but actually are already at the upregulated level, even in the control conditions, suggesting that the tolerant variety is potentially primed for a change in temperature and can more rapidly respond to that because it does not necessarily need to additionally phosphorylate the target proteins. And we have similar things, or kind of other responses where we see that there is a downregulation in the sensitive variety, and this is basically unchanged in the tolerant variety. So this is kind of an idea where we try to identify these markers in multiple varieties, which then could be used in breeding programs. So now kind of switching gears a little bit from this kind of more larger omics approach to kind of really zoom in on a number of candidates that are really, as I mentioned in the beginning, really conserved, which is kind of our prime interest. So we did the same kind of analysis on Arabidopsis, on wheat, and on soybean, where we exposed these seedlings to 1 hour of elevated temperature. And then those that were changing in all 3 species, we call our targets of temperature or TOT proteins. We're initially interested in those that, of course, had kinase activity, but since at the time very little to nothing was known about membrane-associated proteins involved in temperature signaling, we preferred those that were predicted to be membrane localized. And this narrowed it down to basically this one candidate here, which we called TOT3. So it has a kinase domain. This is the phosphosite that we identified as being differentially regulated by temperature, and it has this large intrinsically disordered region. Now, TOT3 is part of a MAP4 kinase protein family. So it's a class of proteins that is conserved throughout multiple species, not only plant species, plays an important role also in yeast signaling, for example. 10 members in Arabidopsis, very conserved in the green lineage. And we were able to identify 3 large clades, and then this kind of third clade we could divide in 4 subclades. And TOT3 is positioned here. And so MAP4Ks are sitting upstream of this kind of canonical MAP kinase signaling pathway, which is made up of MAP3Ks, 2Ks, and 1Ks, and is kind of regulated here by either a receptor or by an upstream signal, and is feeding into this MAP kinase signaling cascade, or it has direct other targets. And this has been characterized reasonably well in yeast and human signaling, where it plays an important role. And the picture is gradually growing also in plants, where it's basically involved in pathogen signaling, developmental processes, light signaling, growth signals, where we are gradually trying to fill the gaps downstream of these MAP4 kinases. So TOT3 is a MAP4K as well. So when we then looked at the loss of function line, and we kind of identified these 2 Arabidopsis mutant alleles, where you can immediately see that at elevated temperature, hypocotyle elongation, this is quantified here, is very much reduced. Also, the angle and the lengthening of the petiole is not as apparent in our 2 mutant alleles, but very striking, and this is what kind of triggered my interest when the PhD student that performed this project, Dai, showed me these data, is that under control conditions, there is no obvious difference, in contrast to a number of other temperature signaling components that also under 21 degrees display a clear phenotype. So this is kind of, at the time, was indicative that this might be a more temperature specific regulator. And as I said, we are looking for conserved regulators. And this is the reason why we identified TOT3. So we also wanted to evaluate this, and we were able to identify tilling alleles with the premature stop codon in the kinase domain that are likely loss of function lines. And indeed, when we compare these tilling alleles, and the same applies for all 3, with the control, where we see a nice, for example here, increase in leaf sheet length at elevated temperature, we see that this is largely lost in our mutant alleles, and this is also quantified here. So indeed, the role in promoting growth at elevated temperature is conserved in the TOT3 mutant across multiple species. But then, of course, there was nothing known about this MAP4 kinase, so we wanted to figure out what would potentially be sitting downstream of this. So for that, we looked first of all in a TOT3 mutant phosphoproteome, and there we identified a number of differentially phosphorylated sites, so proteins that were less phosphorylated in this mutant background, in this kinase mutant background. And to then narrow these down to putative direct targets, we also performed protein-protein interaction assays. So we did tandem affinity purification and co-immunoprecipitation in seedlings, and this gave us 2 data sets of direct interactors, and a number of these overlapped with the phosphoproteome. And these 2 we refer to as TOT3 interacting proteins, or TOIs, and we confirmed this interaction in yeast for TOI4 and TOI5, which are the 2 that sit here in the middle. But interesting, and to some extent also to our disappointment, these 2 proteins are also members of the MAP4 kinase family. So this really didn't get us any closer to what these MAP4 kinases would be controlling in growth responses. Nevertheless, these proteins play a role in hypocotyle elongation. So again, if we look look at our TOT3 mutant, reduced hypoglottal elongation at high temperature. The single TOI4 and TOI5 mutants don't really show an obvious difference. The double TOT3 TOI4 has an additive phenotype, which is indicative that there is partially overlapping signaling between TOT3 and TOI4, but that there are also distinct signaling components. The TOT3 TOI5 double mutant is very much the same as the TOT3 single mutant, indicating that this is likely acting in the same pathway. We have some additional data that support that as well. So this basically led us to the following model, where essentially at high temperature you have this Phi-B PIF4 presbyosteric signaling pathway that impacts on temperature-mediated growth. This had been fairly well characterized, and we had some indications that the TOT3 signaling would act on the activity of the BZR1. We have some new data, which I will show in the second section of my presentation, that seem to indicate that potentially something else is happening, as TOT3 is targeting a protein that might explain some of the phenotypes that we observed. So we now have this module where TOT3 and TOI4 directly interact with each other, have their kind of distinct and common targets. One of these targets and interactors is TOI5, and this feeds into how elevated temperature is mediating hypoglottal and other growth processes. Now, the next thing that we were interested in, and this is one of the things that comes back to multiple stress conditions which are obviously occurring out there in nature. And one of these typical stress conditions that very much co-occurs with heat is obviously drought. So we have reduced precipitation, reduced humidity in the soil, and of course this is in part associated with elevated temperatures. So if you have these kind of conditions, then the plants need to be able to deal with this in the correct way and to adapt their responses. Because if you look at these stress conditions, and this is not only applying to abiotic but also to biotic stress on the level of the stomata, for example, where heat will lead to open stomata to promote cooling. In contrast, drought will lead to closed stomata because you want to prevent water loss. And in addition, biotic stresses will also lead to closed stomata because you want to prevent the entry of, for example, pathogens. Now, what happens if you have both of these stress conditions occurring together? Then you have a clear conflict. Do you open or do you close your stomata? And we were triggered into exploring this in a little bit more detail because we noted that TOT3, our MAP4 kinase, is also expressed in these guard cells. So we wondered if our TOT3 mutant would also have a guard cell stomatal opening phenotype. And indeed, if we look at the TOT3 mutant in Arabidopsis, we see that upon elevated temperature, there is an increase in stomatal opening, and this is associated with an increase in stomatal conductance. In the TOT3 mutant, stomatal opening at elevated temperature is largely abolished, and this is associated with a loss of, or kind of a lack of change in stomatal conductance. And we can indeed nicely complement this phenotype with translational fusion. In addition, we see the same response in our wheat mutants. So we have in the control stomatal opening, quantified here in our wheat tilling lines. We see that this response is lost, both on the level of stomatal opening and stomatal conductance. So clearly indicating that also this response is very much evolutionarily conserved. So we were then wondering what is controlling this stomatal opening downstream of TOT3. So we went back to our TOT3 phosphoproteome, and there we observed that phosphorylation of AHA1, the proton pump, at this penultimate residue, which is important for the activation of the proton pump, is reduced in the mutant, indicating that indeed TOT3 might phosphorylate or might lead to the phosphorylation of the proton pump. This was further supported by the fact that if we co-express TOT3 in tobacco together with the proton pump, we see an increase in phosphorylation at the same position in comparison with a kinase that TOT3 kinase. And we also observe that proton pump activity is increased at elevated temperature, and this is lost in our mutant background. Indeed, indicating that TOT3 is impacting on the proton pump, and this has an impact on its activity. So we then explored if this would indeed be a direct effect, and indeed we could demonstrate that TOT3 GFP is directly interacting with the proton pump. So in transient co-expression followed by co-IPs, we can show a direct interaction. And furthermore, we can also show in vitro that TOT3 is directly phosphorylating a fragment that includes that residue. So if you have here the presence of TOT3 in this autoradiogram, this is autophosphorylation. If we then incubate the proton pump AHA1 fragment together with TOT3, we see nice phosphorylation. If we then mutate this site into an alanine in the presence of TOT3, we lose this phosphorylation at this position, indicating that TOT3 is directly phosphorylating the proton pump at that position. And this was furthermore supported by the kind of genetic evidence that if we use this OST22D mutant, which is a constitutively active AHA1, that is uncoupled from its phosphorylation. So it doesn't need phosphorylation at its penultimate residue to activate proton pump activity. This leads to open stomata, and at high temperature, we can basically rescue our TOT3 mutant phenotype, suggesting that indeed the lack of proton pump activity is responsible for the loss of stomatal opening in the TOT3 mutant. So this kind of led us to the following model, where heat is activating TOT3. This is leading to phosphorylation of the proton pump, and this is impacting on stomatal opening, and this will facilitate plant cooling. But of course, the question that now remains is, what is happening under drought conditions? So under drought, basically you activate ABA biosynthesis, and this leads to the activation of this OST1 kinase, which results in stomatal closure to retain water. So we observed that TOT3 is directly interacting also with OST1. And again, transiently in tobacco, we could demonstrate this direct interaction. And furthermore, both TOT3 and OST1 are nicely co-expressed in these guard cells. So indicating that indeed they can interact in guard cells. What we also noted is that the heat-activated TOT3, which you can see here, is kind of an increase in TOT3 autophosphorylation. So it's kind of indicating increased activity at a high temperature, quantified here, that this is repressed by ABA. So ABA, downstream of drought, is repressing this activity. And we also see the same thing occurring if we have drought present. So basically, under well-watered conditions, there is an increase in TOT3 activity at elevated temperatures, and in the presence of drought, this is no longer occurring. So suggesting that indeed both ABA and drought are leading to the inactivation of TOT3. So we then wondered how this would happen. And for that, we co-expressed OST1 and TOT3 in tobacco with or without ABA. So we were looking for TOT3 sites that were regulated by both ABA and OST1, which would be indicative of ABA activating OST1, and this then leading to TOT3 phosphorylation. And there we identified 3 sites, in addition to a number of other ones, that clearly corresponded to this pattern. So not really obviously or strongly regulated in the mock conditions by ABA or OST1 alone, but the combination of the two leading to increased phosphorylation. So therefore we focused on these 3 sites for further analysis. So first of all, we wanted to confirm that phosphorylation of OST1 occurred on these 3 sites directly, and then we again went for in vitro phosphorylation. So if we co-incubated OST1 with the TOT3 fragment, including those sites, we see nice phosphorylation of that fragment. If we then mutate these sites, and we mutated in addition to these 3, 2 additional ones to this 5A TOT3 protein variant, we largely lose, or we nearly completely lose, all phosphorylation on the TOT3 fragment by OST1. So suggesting indeed that these 3 sites are directly targeted by OST1. So we then next wondered what is the role of phosphorylation at these positions. So for that, we on the one hand used our alanine mutants, so we kind of mutated these sites, so we prevented phosphorylation at these positions, and we also used a phosphomimetic to kind of at these 3 positions to really see what would happen if you would mimic phosphorylation. And a little bit to our surprise, but of course in support of our model, we observed that while the wild type TOT3 variant can nicely complement the response to elevated temperature with respect to stomatal aperture compared to the TOT3 mutant, if we have our alanine mutant, 2 independent lines, this one also nicely complements the phenotype, suggesting that the TOT3 alanine mutant variant is still active. In contrast, if we have our phosphomimetic, we are not able to complement the mutant phenotype, indicating that our TOT3 phosphomimetic variant is no longer active. And this is further supported by our in vitro kinase assays, where if we focus on, then this is TOT3 autophosphorylation, first, if we have TOT3 wild type, there is a certain level of activity of autophosphorylation. If we mutate these 5 residues, we actually increase TOT3 kinase activity reflected in autophosphorylation. However, if we have a phosphomimetic, we largely decrease the kinase activity. In parallel, if we then look at the direct target of TOT3, this proton pump fragment, we see a similar picture. So we have active TOT3 phosphorylating the proton pump. If we have the mutant variant, more active, more phosphorylation on the proton pump. If we have the phosphomimetic, less activity, less phosphorylation of the proton pump. So this is kind of suggesting that indeed OST1 is phosphorylating TOT3 and inactivating it. And this is also reflected in the proton pump activity, which is elevated under control, in a control background, which is not elevated in the TOT3 mutant, but our 5A protein variant is activating, even at control temperatures, the proton pump, further supporting that this is highly active. In addition, if we then look at the sensitivity to ABA, so ABA would lead to stomatal closure, what we see nicely here in our control conditions. If we prevent the phosphorylation at this position by OST1 in our mutant variants, we see that we get a reduced sensitivity with respect to stomatal closure in this genetic background, really indicating that to obtain stomatal closure, inactivation of TOT3 is required by phosphorylation at these positions. And then we of course questioned to what extent is this also present under natural conditions. So if we then look in seedlings and we expose these seedlings to either ABA or drought, what do we see with respect to phosphorylation? So as I've told you, we identified these 3 sites that were very much regulated by ABA and OST1 together. So if we then perform a phosphoproteomics experiment where we expose our seedlings to either control temperature, elevated temperature, or ABA, we basically see that TOT3 at this position is less phosphorylated at elevated temperature. Because at that position you want to have, at that condition you want to have an active TOT3, so you have reduced phosphorylation, which leading to more activity. In the presence of ABA, however, we get more phosphorylation at this position, indicating that this is likely inactivated by ABA. And we see the same picture emerging under drought conditions. So we have elevated phosphorylation at this position under drought, and this phosphorylation is controlled by OST1, because in the OST1 mutant background we see a clear reduction in phosphorylation at this position. So then, of course, the final question was, to what extent does this play a role under drought conditions and combined stress conditions? So if we look at seedlings under drought conditions, we see that wild type, this is basically the complemented line corresponding to the wild type, is rapidly wilting. Our TOT3 mutant, which has more closed stomata, therefore loses water less, essentially wilts less and slower. If we have our complemented line with our phosphomutant variant, we basically see that this is still active as it recapitulates the wild type and wilts rapidly. Our phosphomimetic, on the other hand, which is inactive, recapitulates the TOT3 mutant phenotype. And under combined stress conditions, we see that phosphorylation at these positions becomes really limiting. Because if you then use leaf temperature as a proxy for stomatal opening, you essentially have a change in leaf temperature upon combined stress conditions. So this is kind of indicating that under combined stress conditions, phosphorylation at this position becomes crucial. Because, and this is then the model that we ultimately came up with, where you have heat activating TOT3, controlling the activity of the proton pump, leading to stomatal opening and plant cooling. In contrast, when you then have drought kicking in, leading to ABA levels that are increasing, the activation of OST1, this leads to the inactivation of TOT3 by direct phosphorylation to prevent that this pathway is active, and then you prevent stomatal cooling. And it appears that under drought conditions or combined stress conditions, this pathway wins out and then prevents stomatal opening to really retain water as much as possible. So this is kind of a model that we came up with, starting from a phosphoproteomics screen where we identified TOT3, and we then kind of used iterative phosphoproteome analysis to really map the very specific phosphorylation events downstream of these different factors. And this is kind of the small signaling network that we are currently further exploring. Interestingly, as I mentioned before, we previously, in the context of hypocotyl elongation, positioned TOT3 upstream of breast-steroid signaling, and specifically of the BZR1 factor. Possibly the reduction in hypocotyl elongation that we see could also be due to, of course, the regulation of the proton pump, which is also involved in cell elongation responses. So as take-home messages, I just want to finish by saying that we can use temperature gradient phosphoprofiling to identify clear signaling hubs, that we can use comparative phosphoproteomics, so kind of comparing different varieties with different sensitivities to environmental stresses to identify putative breeding markers. That we can use this comparative phosphoproteomics across different species to really identify conserved signaling components, and that our TOT3 OST1 AHA1 signaling modules integrates different environmental cues to lead to an optimal response under single and combined stress conditions. So with that, I want to just thank the people that were involved in the work that I presented. So the initial TOT3 work was done by Lamdai Vu, a very talented PhD and postdoc in the lab, in collaboration with a number of other people, not in the least the lab technician Brigitte. The wheat work that I showed is mainly the work of Cassio during his PhD, and with the help of Ting Ting during her PhD, and at the moment her postdoc in my group. And then the TOT3 OST1 AHA1 signaling module was the work of Shang Yu, together with Dai and a number of other people that helped out with different aspects of the project, not in the least group of Martin and Pedro, in the context of trying to reveal these intricate signaling mechanisms and the physiological analysis associated with this. So with that, I just want to advertise our plant PTM viewer, which is kind of a resource that we developed a while ago, and that was kind of further expanded on by the group of Frank van Breuzegem, specifically Patrick Willems, to incorporate not only phosphorylation, but a number of other PTMs in a wide range of plant species, kind of making this an easily searchable tool to look for your favorite modification and your favorite protein. And we are currently working on an update of the plant PTM viewer, the 2.0 version, that has a number of additional features. So stay tuned for that. This is freely accessible for everybody, and it gets regularly updated with new data sets. And finally, we're at VIB and in the department, we are always open for people that are interested to apply for a postdoctoral fellowship. So do not hesitate to contact me or our grants office if you would be interested in a postdoctoral fellowship or a PhD in our center. So with that, thank you very much for your attention, and I'm happy to take questions.
So thank you for delivering this great lecture. I guess you can close the presentation now. It's pretty much amazing. I'm always very impressed with the amount of data you generate, with the amount of results you generate. And one of the things that I like most is how you move right from discovery to real functional analysis. This is so cool. This is pretty cool.
Yeah, this is really the thing that we wanted to do. Yeah, that's kind of not to just kind of capture the change in phosphorylation, but really try to use this function element of functionality as well.
Yeah, absolutely. So I have one question, maybe the very first one. Do you have any experience in translational science, like in doing field work or using phosphoproteomics to understand how plants respond in the field?
Yeah, so we also generated the data set in wheat, where we kind of exposed wheat plants, more mature wheat plants, to elevated temperature, and there we kind of also looked at the responses in the leaf, but also in the anthers, for example, because these are also the pollen in the anthers are also highly sensitive to a change in temperature, and that's a data set that we are still currently exploring. That's in wheat. we also had a pretty big project together with Ross Sazani and Anna Locke on soybean, where we basically put soybean in grow chambers, expose these to elevated temperature, and also cold, kind of lower temperature. But in addition to these kind of grow chamber experiments, we also did that in the field. So we basically put soybean in the field, exposed these to elevated temperature, and then looked at changes in phosphorylation, but also a number of developmental yield and physiological parameters. And we also did that with respect to cold, where we put like multiple varieties in the field, and we basically used the natural cold environment by planting or sowing our seeds a little bit earlier than the usual time. So we're trying really to also bring this kind of growth chamber knowledge to the field, and then to see does it correlate or not. And we're currently analyzing those data to see if there is indeed a correlation, or if the responses are completely different, if you of course have additional environmental factors impacting on the phosphoprotein.
Yeah, great. This would be the ideal situation, right? From discovery to translational, right, science. And this would be my second question about the cold stress. You know, it seems that TOT3 plays a central role in signaling. So what do you expect it to really respond to cold stress, or CO2, like the stress that I pretty much used to work with?
Yeah, so that's something that we're checking at the moment as well. So we identified in wheat that the TOI4, TOI5 module, which is then part of this TOT3 complex, is kind of maximum in its activity at the lower temperatures. And we also had a number of observations previously that suggested that TOT3 might indeed play a role across the complete temperature gradient. So far, we've mainly focused on the elevated temperature. We also kind of are now looking a little bit in more detail in what happens at lower temperature, but we don't have any clear-cut results there yet.
Okay, good. Thank you. Great. Let's just wait for questions. Maybe somebody has a question. Adriana?
Thank you, Thiago. Thank you for the nice presentation. Very interesting, and also the way you translate your discovery results is very amazing. I'd like to ask you if you can to give us more details about your decision of the phosphoproteomic enrichment. What method that you use, and why do you prefer this method? Can you please comment about that?
Okay, yeah. So when we started, we went for a number of options, and I wanted to have like an experimental setup or methodology that would be extremely versatile, because we already had indeed the ambition to explore this in multiple plant species, multiple varieties, but also with kind of complex experimental setups with multiple conditions and multiple time points. So we really wanted to focus on label-free mass spectrometry, because that would indeed increase or would not limit us to the number of samples that we could analyze. And then of course we enrich for our phosphorylated proteins, phosphorylated peptides specifically. Now, so following the tryptic digest, we kind of landed on these titanium IMAC beads as being the best ones in our hands to use. So we get this, I think it's a South African company that provides these, and in our hands those work best. There are of course a number of other approaches out there, but we kind of stuck to this for the time being.
Thank you very much. And I also have a curiosity, in fact, if you were able to analyze the global balance between the kinase and the phosphatase in the global proteomics, to check, because some of them are involved in the pathway that you are analyzing, but you also can evaluate their abundance and also their phosphorylation. How did you deal with this data when you consider these temperature changes?
Yeah, so what you're referring to is the abundance in change of the target proteins that are commonly regulated by the kinase and the phosphatase.
Yes.
Yeah, so that's a tricky thing. I mean, you always look at the end result, no? So you kind of see the amount of phosphorylation that is a balance between kinase and phosphatase activity. One of the assumptions is, of course, that if you have, for example, in the context of temperature, TOT3 gets activated, and then the downstream components will be phosphorylated. That at the same time the negative regulation by the phosphatase would need to be inactivated, so that you kind of so that kind of helps us as well to look for potential substrates that have a kinase and a phosphatase, because the regulation of the kinase phosphorylation or the phosphatase phosphorylation themselves is indicative for their activity, depending on where the phosphorylation is. And this is the kind of information that we're also using in our network analysis, for example. So if you go across time and you see that the profile across time is kind of an upregulation of kinase activity, that this is then mirroring what you would see in the phosphorylation status of the substrate. Yeah? So you have, this is the profile of the kinase, increased activity, so your substrate should more or less follow the same profile. If you then have a phosphatase that gets activated and you see this kind of profile of the phosphatase, then your substrate should follow the opposite curve, essentially. So that's kind of what the way we are trying to leverage that information to see if indeed if there is more of a kinase or more of a phosphatase that is active. And then of course the next step is what happens in the mutant backgrounds. So if we then knock out the phosphatase, knock out the kinase, do we then see indeed that the expected changes occur? So that I don't know, does that more or less address your question?
Yes, there is. Yes, thank you very much. Mm-hmm.
Well, if anybody else has questions, I do have one. Adriana, you want to say something?
I also
enjoy
this opportunity.
You can do it first. Sure.
Okay, thank you very much. Eve, what about when you have the protein, you have the peptide, phosphopeptide enriched, but you do not have the non-phosphorylated peptide enriched. So how do you deal with this data? Because you analyze the global proteome and also analyze the enriched peptide, and not necessarily you identify the protein in the global proteome. And how did you?
Yeah, that's one of the things that we try to address very much in the beginning when we started our analysis, where we had this kind of goal to try and normalize the kind of change in phosphopeptide abundance against the change in protein abundance, because obviously your change in phosphopeptide abundance is not only because of kinase and phosphatase activity, it also depends on the amount of protein that is there. So if there is a kind of increase in translation or an increase in degradation, that will also of course impact on the overall protein abundance and also then the phosphorylated amount, which will then be reflected in the phosphopeptides. So the way we kind of tackled that in the beginning is indeed in parallel on the same sample, analyze the phosphoproteome and the proteome, and then try to normalize for this. This then really allows to more or less identify those phosphopeptides that are to a large extent purely regulated by kinase and phosphatase activity, because all the rest you remove. The downside of that, well, there's basically two elements that we ran into as main reasons why we stopped doing that. One is, of course, if you then have your phosphorylation playing a role in protein abundance and in either degradation or trafficking or something like that, you would filter out those very interesting candidates. So that's one of the reasons. But the major reason is essentially that with your phosphoproteome, because you have this enrichment step, you also kind of capture lowly abundant proteins, which you do not capture in your overall proteome. So many of the phosphopeptides we could not normalize because we didn't capture them at the time. So we were running 2MS analysis, and we could not kind of use it to the extent that we wanted to. So therefore, we now kind of, in most cases, we stick to just doing the phosphoproteome, and then we have a number indeed of false positives that we then see popping up later on in our subsequent validation analysis. So it's a bit of a trade-off. I mean, if you want to be really, really, really sure about your candidates, then you can indeed do this proteome normalize. If you have a little bit of more flexibility and you say, okay, it doesn't matter if I have some false positives and I'll notice in the downstream analysis what is happening, then it's not necessary to include that. I think now maybe we can get a little bit closer with for example, using DIA, which kind of has a better coverage. But we haven't explored that yet.
Okay, nice, thank you.
I have a question from the audience. Can you read it? Can you read it, Yves?
Yeah, so what we mainly have been using is MaxQuant. So this is for most of our analysis, this is what we have been using. Recently, also for a number of other projects, we started to explore FragPipe as an alternative. One of the reasons why we used MaxQuant in the beginning is it was available, freely available. It works very well. It's rather user friendly, so I think with limited training you can go for it. I think FragPipe has similar features, so I think that's also a very valuable search engine to use.
Great, thanks, Yves. I don't know if we do have more time for that, but I was wondering if you would say a few words about the ART Genetics project that you do have. That's because I like it so much. So,
yeah, so I did
say something about it.
Yeah, so I didn't prepare a slide for that, but
it's okay.
But this is something that I started a number of years ago with a friend of mine. So we were kind of wandering in a museum. So he's an art historian. So I'm kind of a plant biologist, geneticist, molecular biologist, and we thought, okay, are there ways where we can combine our expertise and our interests? And we were discussing at a painting if this vegetable, this fruit that was depicted there, if it really looked like that in the 17th century, or if this was just a bad painter. So we then kind of trying to figure out what was happening. Were there purple and yellow carrots in the 15th and 16th century, or were they all orange, or when did the orange carrot appear? When do we start to see modern strawberries? So the idea behind this is that we try to capture diversity, domestication processes throughout history by looking at artwork. And you can also see this, and a nice example is wheat. So if you look at wheat being depicted, for example, with the old Egyptians 3,000 years ago, you see wheat is the size of a person. If you look at Bruegel, one of his very famous paintings, he paints wheat the size of a person. If you now go into a modern wheat field, it's basically just above the knee. So this is kind of a modern change associated with the Green Revolution and Norman Borlaug. So the idea is a bit, do we see this change across history by looking at art, and with all the knowledge that we have now on processes, developmental processes, physiological processes, taste, color, shape and size, and these kind of things, the availability of the genomes, do we also have the molecular explanation for these differences? And this is something that we try to do as a kind of a hobby outreach project to kind of expose people to the basics of genetics, the basics of how to look at art. So we have people that are interested. We have a couple of publications on this, but we also have an Instagram account which is called Art Genetics. So if you look for that, you'll find a number of these little stories and pieces of information there on that topic as well. So I'm always glad that people are excited about this. the thing is that, so I've been doing what I think is cool, very fundamental research for like 20 years now, looking at asymmetric cell division, signaling processes, root architecture, heat stress, and these kind of things. And there was always very limited interest by the general public and by the media. Since I go to a museum with a friend of mine, we've been all over the place. We've been in quiz programs, television, podcasts, newspapers, nationally and internationally. So this is really something that attracts the attention of people. So we kind of also then use it as something to leverage, exposing them to kind of the basics of genetics.
Yeah, this is just a great outreach project. So I'll check it out, in the Instagram.
Yeah.
I always like to
I recommend people to check it out.
Yeah, tell people what you do. And our genetics just amazing. Great, Eve. Thank you. So I guess we don't have any more questions. We are running out of time too, right? So again, thank you so much for coming to this section. It was great to see you again. Adriana, do you want to say something about this is the last meeting we have, right, for the year?
Thank you very much for joining us in this edition. And like to thank you very much for the opportunity to be the host together with Thiago in this 5th edition. And thank you very much. Thank you, Thiago and also Giuseppe, who invited us to be part of this, my friends MASSA. And thank you all for joining us every Friday in this last month.
Good. Thank you very much.
And thank you, Adriana. It was great. It was a pleasure being here. See you guys.
Thiago, see you.
See you. Bye-bye.